Regulation of glycolysis and glucogenesis, the role of Aldolase expression. Welcome, everyone. Today we'll explore how cell balance energy use and storage through two major pathways. The first pathway is glycolysis and the other one is gluconeogenesis. We will also see how one enzyme, Aldolase, sits at the center of this regulation. By the end of today's lesson, we will be able to compare glycolysis and glyconeogenesis and describe Aldolase's role in both pathways. We will then describe how Aldolase isoforms and their tissues are distributed. Lastly, we will explain how transcription factors such as HNF1, HNF3, CEBP, and DBP regulate Aldolase B. Before we go into this lesson, take a look at your worksheet that is linked in the slideshow. What I need for you guys to do is copy the definitions and the example that will help you remember. So, isoforms are a different version of the same protein that happen in different tissues. If you were to think of an example, think of isoforms like different flavors of the same enzyme that is made. The second is transcription factors. This protein turns genes on or off by binding to DNA. Transcription factors are like managers who decide when the enzyme workers should start. Lastly, we have expressions. This is the process of making a gene product. If a gene is being expressed, the cell is actively using it. Now, before we dive deeper, let's activate what you already know. Glycolysis might sound complex. It's a big word, but you actually have studied parts of it before. This is the pathway that helps turn glucose into energy. So, what do you guys already know about glycolysis? You're going to answer in your worksheet. Also, when do you think this pathway is active in the body? Do you think it's during eating or do you think it's during fasting? Great thinking, everyone. You've already recognized the energy pathways turn on and off depending on your body's needs. Next, let's review how glycolysis actually works and why the cells need a separate pathway to build glucose when energy runs low. Now that's called glyconeogenesis. Let's take a step back before we get into the molecular details. Think of your body like a bank account. You can either deposit money or withdraw money, but doing both at the same time would make no sense, correct? The same logic applies to your cells. They have to regulate when to use energy and when to store it. First, let's visualize this. Glycolysis is like spending money. It connects scientifically because glycolysis is the breaking down of glucose for ATP. This happens when you need immediate energy. For glyconeogenesis, it is like saving money. This pathway is building glucose for later use. This happens during fasting or low energy intake. Lastly, regulation. This can be compared to budgeting wisely, as it helps to prevent energy waste. Now, if both glycolysis and glyconeogenesis ran full speed at the same time, the cell would be like someone transferring money between two accounts and losing fees every time. That wouldn't be fun. This is wasting energy as heat instead of producing anything useful. Now, when you eat and glucose levels rise, your body tips towards glycolysis, breaking down sugar for quick ATP. When you have eaten, the balance shifts towards glyconeogenesis, rebuilding glucose to keep your body's sugar steady. This constant building act is how your body maintains homeostasis. I'm going to take a pause, and then we'll give you guys time to answer checkpoint question number 2. What might happen if the cell did not regulate these pathways? Think about what would happen if they both stayed active. Write a short hypothesis in the box. Remember, a hypothesis is an educated guess. Now that we have understood why regulation matters, let's closely at how glycolysis actually works. The pathway that turns glucose into energy. Now, as we explore, pay attention to where the cell invests and gains ATP. These are called the control points of the system. I'm going to give you guys a few minutes if you have not completed the checkpoint question. All right, let's move on to glycolysis overview. As you see, we have an image of what glycolysis looks like. So, now that we know why the body regulates energy pathways, let's look at one of the most important ones, glycolysis. This pathway happens in every cell in the cytoplasm. This is how our body gets quick energy from glucose. This is known as a this process is universal, and this means that it happens in nearly all organisms from bacteria to humans. This is why biochemists call it one of the most ancient metabolic pathways, as it's a survival mechanism that cells have used for billions of years. As you can see in the image, the cell invests ATP to prepare glucose for breakdown. This is spending money to make money. Later, the pathways pay off with a net gain of two ATP molecules and two NADH molecules. So, let's walk through glycolysis in two halves. The first half costs the cell energy. This is where you have to spend two ATP molecules to prepare glucose. In the second half, the cells earn energy back. This results in the net gain of two ATP molecules, which is like your body's pocket change for quick energy. In the next slide, you will be able to see each step in the three important irreversible control points: hexokinase, PFK1, and pyruvate kinase. All right, so the steps in glycolysis is known as the investment and the investment payoff. So in phase one, this is where we have the energy investment. The first step is the irreversible hexokinase. This is where glucose is converted to phosphorylate to glucose 6 phosphate. This is important because this traps glucose inside the cell, preventing it from leaving once it has phosphorylated. The second is phosphoglucose isomerase. This happens when G6P is converted into fructose 6 phosphate, which is also known as F6P. This matters because it rearranges the molecule for the next phosphorylation. The third step in the investment process is the phosphofructonase 1. This is known as the PFK1. This is the second irreversible step and the main point. This is where another phosphate is added, creating fructose 1,6 biphosphate. This is important because this is the committing step. One here, glycolysis must continue. The fourth step is Aldolase. This is the fructose 1,6 biphosphate, which slips into a 3-carbon sugar known as G3P and DHAP. This is important because this is where the 6-carbon glucose becomes a 2,3-carbon piece. The last step in phase 1 is triphosphate isomerase. This is where DAP or DHAP is converted into G3P. Both molecules continue identically. This ensures that energy production happens evenly from both halves. Before we continue with the next phase 6 to 10 steps, let's have a mini checkpoint. So this is where you guys get to take a pause and you guys can go back into the lesson and figure out what you have missed. So for a mini checkpoint question, what step do you think that locks glucose inside the cell and commits it to glycolysis? All right, now let's move to phase 2. This is where the energy is paid off. The goal of this phase is to create ATP and NADH. Now we have our sixth step. Our sixth step is called glyceraldehyde 3-phosphate dehydrogenase. This is where each G3P is oxidized to 1,3-biphosphoglycerate, which forms the NADH. This step is which harvests high energy electrons for later ATP production. The next step is phosphoglycerate kinase. This is the transference of a phosphate from 1,3-biphosphate to ATP and 3-phosphoglycerate. This is the first ATP payoff step substrate level phosphorylation. As we are getting closer to the end, the eighth step is phosphoglycerate mutase. This is where the 3PG is rearranged to 2-phosphoglycerate. Here is where the molecule is preparing for dehydration. Now, the ninth step is where Aldolase is created. Water is removed from the phosphoenolate pyruvate. This is important as it creates a very high energy intermediate. The last step is known as pyruvate kinase. This is the transfer of phosphates from PP to ATP in pyruvate. This is the final energy release and pyruvate formation. This step is also an irreversible step. All right, so now that we have explored glycolysis and how cells break down glucose for energy, let's flip the system and talk about how the body builds glucose when it's running low. This process is called glyconeogenesis. This just means the creation of new glucose. Now, if you can remember, glycolysis happens in the cytoplasm. Glycogenesis happens mostly in the liver and the kidneys. This is due to the fact that these organs act like the body's energy managers. When you're fasting, asleep, or exercising intensely, they rebuild glucose to keep your body blood sugar stable. Now, unlike glycolysis, which makes ATP, glycogenesis spends ATP and GTP. This is an energy investment. Think of it like charging up your savings account. You are using resources now to make sure you have glucose for available later. As you can see in the image, both pathways are mirror images of each other. However, they don't happen together. When one is turned on, the other is turned off. This prevents the cell from wasting energy cycling back and forth. Now here's a mini checkpoint. What might happen if glycolysis and glycogenesis ran at the same time? Take a few minutes and answer the question. Now, you have also learned that glycolysis has three irreversible steps: hexokinase, PFK1, and pyruvate kinase. Glycogenesis uses different enzymes to bypass these steps. These bypass enzymes are the cells that work around to reverse the process safely and efficiently. The bypass enzymes include glucose 6 phosphate, fructose 1,6 biphosphate, pyruvate carboxylase, and PAP carboxyl kinase. Now let's look more closely at how this regulation works. We'll see how molecules like ATP and AMP serve as signals to tell the cell which pathway to activate. I know some of you are wondering what this chart means, so let's make sense of it, okay? So this chart shows how three major regulators control the direction of glucose metabolism. You can think of this like road signs at a metabolic intersection that tells the cells which road to take. One road heading towards glycolysis, breaking down the glucose for energy, and the other one heading toward glycogenesis, building glucose to store energy. If the cell has a high ATP, that means it's full of energy, so it does not need to make more. High ATP will slow down glycolysis and activate glycogenesis so the body can store or rebuild energy. On the other hand, if the cell's ATP is low, AMP levels go up. That's the cell's distress signal saying we need more energy, so AMP activates glycolysis and shuts off the glycogenesis. Sorry, let me repeat that for you guys. So, when the cell distress signal is saying we need more energy, the AMP activates glycolysis and then shuts off glycogenesis. Now, as for fructose 2,6 biphosphate, this is known as the master switch. This molecule is the most powerful controller. It activates PFK1 to speed up glycolysis, but stops fructose 1,6 biphosphate to slow down glycogenesis. It is like a molecular light switch that can toggle between using energy and saving energy. Again, we're going to have another checkpoint, so please take a few minutes to answer this question. I'm going to also give you a few minutes to get caught up on your worksheet. So for the checkpoint question is, why do you think low ATP activates glycolysis? Now that we have covered how these pathways are controlled, let's focus on a single enzyme that plays a role in both, and this is known as Aldolase. Again, let's take a few minutes. All right. I'm glad I was able to give you guys time to get caught up. So let's look at Aldolase. All right. So here's where things get interesting. The enzyme Aldolase acts as a bridge between glycolysis and glyconeogenesis. It is one of the few enzymes that works in both directions. This reaction is reversible. This means that it goes either way, depending on the cell's energy state. When the energy is low, the reaction moves forward for glycolysis. When the body needs glucose, it reverses for glyconeogenesis. This reaction is like a reversible zipper. It can zip up or down depending on what the cell needs to do. This explains the reversibility concept. Now I want you to pause and reflect. Why do you think reversibility is useful for a cell? All right, now that you know what Aldolase does, we are going to take a closer look at its different forms or isoforms. Remember that word from the beginning? We will explain how each version functions in a specific tissue, like the muscle, brain, or liver. Now, we just saw how Aldolase works as a bridge between glycolysis and glyconeogenesis, but the body uses different versions of this enzyme called isoforms. These isoforms are specialized for different tissues and functions. Each of these isoforms belong to the same gene family, so Aldolase A, Aldolase B, and Aldolase C. Each one codes for a slightly different protein structure, tuned for the energy that it needs of its tissue. If you're having trouble understanding, think of isoforms like different tools from the same toolbox. A hammer, a wrench, and a screwdriver all come from the same set, but each one fits a unique job. Similarly, Aldolase A, B, and C all catalyze similar reactions, but in this tissue best suited for their version of the job. Which tissue do you think would rely most on constant glycolysis, which is a process that is always turned on? Now, let's zoom in on the liver's form Aldolase B. It is the only isoform that plays in both pathways in its role in regulating blood sugar. This makes it incredibly important. So we have Aldolase B. Aldolase B is special because it not only handles glucose, but it also processes fructose, which is another common sugar found in fruits and processed foods. In the liver, Aldolase B breaks down fructose 1 phosphate into usable energy intermediates. As you can see, we have the pathway that it goes. So it goes from fructose to fructose 1 phosphate to Aldolase B to G3P and DAP. When Aldolase B doesn't work properly, can't be broken down efficiently. This causes a dangerous buildup of fructose 1 phosphate in the liver, which ties up phosphate needed for energy metabolism. This results in hereditary fructose intolerance, which is also HF1, which is a rare genetic disorder. With this disorder, many patients experience severe hypoglycemia, vomiting, or liver swelling after eating fructose. The good news is that this is manageable with early diagnosis and diet modification. This is a really great example of how understanding enzymes can save lives. So, if Aldolase B is not doing its job, how does that affect blood glucose levels? Now, let's see how the body controls when to make more Aldolase B through gene regulation. So, gene regulation of Aldolase B. The expression of Aldolase B in the liver is not random. It is tightly regulated by special proteins called transcription factors. These act like an on and off switch that bind to gene promoters region to either start or stop transcription. Let's imagine sorry guys. Let's imagine transcription factors as teachers giving homework permission slips. So let's start. All right, HNF1, you guys are the liver cells, and you guys may begin transcribing your metabolism genes. HNF3, you will help adjust for fasting or feeding conditions. CEBP, you will manage energy balance across the system. And lastly, DPB, you will work with your circadian rhythm, helping to keep your metabolism in sync with your sleep wake schedule. Now, why might it be useful for this liver's metabolic enzyme to follow your daily rhythm? Now that we know the transcription factors that are involved, let's see how hormones like insulin and glucagon glucagon trigger them through signaling pathway. As you can see, this is a pathway that you guys can see and how it follows. The diagram shows how hormones act as a cell messenger system to turn genes on and off. Now let's trace the signal. When the glucan levels rise, for example, during fasting, it triggers CAMP signaling. CAMP signaling activates protein kinase A, which enhances the activity of transcription factors like HNF and CEBP, which increases aldolase B transcription. This turns on glyconeogenesis so the liver can produce glucose. On the other hand, insulin has the opposite effect after eating. It suppresses this pathway because the body already has plenty of glucose. Now we have another checkpoint. I want to know which hormone, either insulin or glycogen, stimulates glyconeogenesis. Now that we know that we've seen how these signals regulate aldolase B, let's connect this back to tissues where each isoform plays a specific role. Now we have tissue expression. So for the liver, aldolase B is for both pathways, while muscle aldolase is for glycolysis only, and the brain is aldolase C, which has a high ATP use. So, just like players on a team, different tissues have different energy priorities. The liver acts as the body's energy hub. It makes and breaks glucose. Muscle tissue focuses on using energy for movement, and the brain requires glucose supply for electrical activity. As stated, we can think of our organs like a players on team. So the liver is going to be the strategist, controlling when to store or release fuel. The muscles are the athletes, burning glucose rapidly for power. Lastly, the brain is the thinker. It is always demanding a steady glucose supply. Now we are going to see how these differences matter clinically, especially when regulation fails. So let's look at the clinical application, which is very important. So without Aldolase B regulation, there will be a sugar buildup known as hypoglycemia. It can also demonstrate the importance of enzyme control, and a real-world connection is genetic testing detects Aldo B mutations. So now, when regulation fails, metabolic consequences can be really serious. As stated, if Aldolase B is underexpressed or defective, fructose and glucose intermediates accumulate, trapping phosphates and depleting ATP in the liver cells. This results in low blood sugar, cellular stress, and even liver damage. This is one of the reasons why geneticists and metabolic specialists use gene testing to de- to detect ADO-B mutations early. Understanding biochemistry is not just academic, it can save your life. Careers in biochemical genetics, molecular diagnostics, and medical research all rely on this kind of enzyme pathway knowledge. So let's wrap this up. You guys did wonderful going through each of the checkpoints and understanding what glycolysis is and glycogenesis. Now, glycolysis and glyconeogenesis are two sides of the metabolic column. One uses the energy, the other stores it. Aldolase is the key enzyme that links both together through regulation transcription factors, which ensures energy balance across tissues. As you can see, this image, it kind of shows the comparison between the two. Here are my references for this presentation. And lastly, here's a link to your worksheet. Make sure you have it completed. You can either do it by hand or you can do it on the internet by making a copy and typing it in together.